Good morning, everyone. Good evening, good night. I'm sure you are all coming from different time zones. So it is my great pleasure to introduce Dr. Lakshmi Narayan Iyer from NCBI that you've been hearing about in the last several days, which is the hub for all bioinformatics, correct? So Dr. Lax, that's how I call him. Graduated from Texas A&M University, and his expertise there during his graduate school was plant viruses. And he's been at NCBI for almost 20 years. And his main focus of his research is protein and domain evolution. Now, Dr. Lax is actually, as you'll all see, one of the most humble, modest person I've ever met, and have the fortunate of meeting and knowing him for 20 years. He has actually made lots of discoveries himself in his career, lots of discoveries of domains. And I think briefly I talked about PFAM domains when we did UniProt hands-on session. A lot of domains there has actually been discovered by Lax. And his research focus is been on many different varieties of different aspects, but everything centered around evolution. So if I were to, think about the best evolutionary biologists, it's all at NCBI, all clustered in one place, and he's one of them. And so it's really a pleasure. And thank you, Lax, for taking the time. I know you have a very busy schedule. Thank you for taking the time to, share what is going to talk about what it takes to make discoveries in silico, right? So because he's made the discovery, so he knows exactly how it works and how you can use actually all the data that we have to make discoveries. There's still room for lots of things that we still need to find. So without further ado, Lax, all yours.
So good morning, everybody. Good morning from my end, that is, and whatever end yours are, good evening, good night. Thank you so much, Sona. I think it was an interesting conversation I had with her before saying, okay, I'll give a talk, because I didn't know the kind of audience that I was dealing with. Scientists are usually, we are more comfortable with talking to a scientific audience, because you know that 90% of them are likely to fall asleep, in the first 10 minutes. So, and you have a certain conversation that you can build upon based on very technical information. So I heard from Sona that a large number of members of this audience, this group, are high schoolers and beyond. So you all are very young. And rather than talking about, the discoveries that I made, or, the kinds of things that are at the cutting edge of our science and so on, I thought I would sort of give you all a very general overview about what it takes to make discoveries in science in general, but actually from a computational biologist viewpoint, or from a theoretical viewpoint, or from a what you may call a bioinformatics viewpoint. What are the kinds of discoveries that have been made? What are the kinds of contributions that bioinformaticians or computational biologists have made to the sciences, and how have they impacted the science? So it's a kind of it's a very general talk, so feel free to stop me anytime you want. And I have no particular direction, but let me start by sharing my screen, and perhaps my slides. Okay, give me a second. Okay. Screen. All right. So Sona, can you see this?
Yes.
Okay. All right. So I thought I would kind of give you an overview of not just what discoveries are out there, because of course there are thousands of discoveries made by computational biologists, but what it takes to make discoveries in silico. In silico, as you know, is entirely computational or from a theoretical viewpoint. But in a sense, it also points to a very general aspect. That is, what type of scientists are out there? And this is a very difficult thing to talk about and classify, because one might think there are many different types of scientists. But in general, many greater thinkers have thought about this. And there's a very famous evolutionary biologist called E.O. Wilson. And he talks about 6 different categories of scientists that are out there. And it's not that we are all clubbed into one category, but we are an amalgamation of 1 or 2 more categories. So a scientist that has all facets of all these different aspects that I will talk about is perhaps one of those Nobel laureates and the person who deeply impacts the science. The remaining of us have 1, 2, or a few of these skill sets, these types of characters that help us make discoveries in the sciences. So my viewpoint would, of course, be from a computational biology angle. Also, the fact that you all have spent some time studying different tools and aspects of computational biology, so you might be able to make some connection. All right, so as you all know, the basis for all this, the basis for computational studies is sort of, more recent in terms of the history of biology, in the sense that up until, say, the early 90s, that was the time when I was a graduate student or when I was in high school, much of biology was conducted in the lab. There were theoreticians who did collect data. For example, much of the data that was collected was on morphological aspects, behavioral aspects, biochemistry of organisms. But it was always limited to a certain set that are typically called as model, or in molecular biology especially. Most of the discoveries that were made before the 90s were all based on certain model organisms. So if you wanted to study bacteria, you have organisms such as Escherichia coli or Bacillus subtilis. If you wanted to study animals, before human models became tractable, there were mouse models for genetics and other animals that were used for different aspects of life, mouse and flies and nematodes. And then for plants, you had Arabidopsis. So the view of biology that emerged was somewhat limited by the information obtained by studying these organisms. And of course, the idea was that what applies to them perhaps generally applies to all different organisms of life, which is somewhat true, but not exactly so. But by the mid-90s, there was this new revolution in biology, which is the ability to sequence DNA. And once on a large scale, people did sequence DNA and before that, but by the mid-90s, we could now get the DNA sequence of entire genomes. And that was a very exciting time. I still remember I was a graduate student then, and I was an experimentalist. I was working on viruses of all things. And I thought, this is something I don't want to miss in life. And perhaps that was and I read the first few papers of how people have been able to sequence the entire genome of an organism, and then they are able to extract information from that genome. It excited me to no end, because I thought, hey, here is something where new discoveries can be made. Because one of the things that became obvious is that when you look at these genomes, you perhaps knew about 7 to 8% of the biology of the organism based on the genes. It was this huge 90% of biology that was yet to be discovered. So along with that, um, over this, I mean, from 93 to now, in the past 95 to now, about 15 years or so, or sorry, it's been, sorry, 25 years or so, the number of completely sequenced genomes have increased tremendously. You know, we have a few, at least 10,000 bacterial genomes and a large number of genomes of eukaryotes. So the amount of information that is out there is tremendous. And you cannot work on every single organism. But yet, how do you, the key thing that came out of this was that you could extract biologically relevant information by studying this large amount of information. At the same time, um, there were many new approaches because of the improvements in technologies that you had various system scale approaches. You may have sort of studied this over the past week about large scale protein interaction or transcription expression databases that are able to crunch large information about everything of so all the genes of an organism all through its life. How is it expressed? Or for that matter, if you take a certain cell population, say you take liver cells and you get all the proteins, how do they interact with each other, and so on and so forth. So this kind of system level information was also there. So it's a lot of information that has been generated and classified and kept in a in silico realm. And as I told you, we know very little about, we knew very little, now we know a little more about life in general. And hence it became a big challenge, and it still remains so, to extract new biology, to make new discoveries. And in reality, if you have what it takes, you can just be sitting at home and making very interesting discoveries with a regular PC. Okay, so the foundation of all this is computational genomics. So there is this in bioinformatics, you have the computer science side, which in parallel to the improvements of technologies also involves building new algorithms, catering your approaches based on the kind of data and also the technologies. For example, now everything is on the cloud, they say, and so on. But on the other end, from a perspective of a computational biologist, there's the biology, and that is perhaps the greatest motivation for many of us in this, which is to try and understand life in general. What can we understand about living systems and life forms? How deep can our questions go? Can we make certain predictions that were not made entirely based on experimental efforts? Can we make new discoveries about behaviors or about biochemistries, or can we make new reagents just by studying these proteins in silico? So there's this whole biology aspect of it. So there is the biology and there's the applications that come out of understanding that biology. And one of the interesting things about biology is somewhat different compared to some of the other traditional sciences, which is physics and chemistry, very generally. And in that, biology is essentially a product of a historical process, and much of evolution has resulted. It's a kind of stochastic process. It is not exactly predictable. You have various possibilities, but only some have been chosen. Now, who was to know that 65 million years ago, a meteor would hit the Yucatan Plateau and then destroy all these large dinosaurs and give a niche for all these little mammals to diversify and occupy various niches. And here we are, one of these products of that event. So many of these events are stochastic in that we can't predict them, and that makes biology very interesting. And from a pure biology perspective, much of biology can be understood if you understand evolutionary principles that, or if you look at systems from an evolutionary viewpoint. I won't dwell on that too much today, but perhaps you will get a flavor for what I'm trying to say here over this talk. All right. So when we talk about the kinds of scientists that are out there, and we have certain archetypes in different fields, and so I said there are 6 categories of scientists, and this kind of categorization is inspired by Leo Wilson's book, which is a very good book for you all to read in high school. It's called Advice to a Young Scientist. A wonderful book, or is it Letters to a Young Scientist? I don't recollect. And also, this came out of discussions with group members. And so the first archetype that I would like to discuss is the so-called technician. And the technician is one who basically accomplishes technically demanding tasks. He has a lot of energy to go through very minute levels of information. And in fact, this aspect of categorization and classification is extremely important for the scientists, or it lays the bed for developing new science at all levels. And just wish to clarify that in these categorizations that I make, I don't think any one type of science or one type of scientist or one type of approach is better than the other. Because what happens is science only develops when you have a well-rounded diversity in the types of scientists and scientific problems that are there. So, the archetype of the technician is Carl Linnaeus, who very systematically looked at various life forms and even developed a binomial nomenclature for categorizing them and classifying them. Whereas this itself didn't mean that he had an idea of the evolutionary theory or he had some great ideas in biology, but it formed a bedrock by which people further down could analyze information. So in terms of bioinformatics, in terms of computational biology, one of the key developments has been the development of various databases. And it isn't a trivial problem because you have to plan in terms of the extent of data that is going to be out there, the kind of tools that are likely to directly port into the easeability, the ease of using these databases. So there's a lot of thought that goes into building these things. So you may have already learned about several databases at the NCBI, and each of them is actually a lot of work in trying to understand how you could represent this information in a digital format that can help us perform further experiments using that information. So in this you have various categories now for sequences of DNA, RNA, and even proteins. You have the NCBI databases, and the NCBI is almost 30 or more years old, maybe 40 years old, and there are at least 100 people who are behind, who are constantly working and behind these databases, so that they provide this information in a way that can be used by others. So the technician actually has a very key role in the development of the science. Then you also have other databases, such as the PDB database of protein structures and molecular structures in general. Then you have databases such as ProCite and PFAM that are involved in using certain, where you can actually, it stores information in a particular way so that you can mine and find homologous relationships either by sequence, or you have databases such as the DALI database that stores structural information. So this is one type of scientist that you'll find, the person who's interested in learning to, typically a computer scientist who's interested in learning to, in putting his information in a format that is very usable, easily retrievable, and scalable. You can make a tool that works, say, for the next 2 years, but can you make a tool or a set or a database that works for the next 20 years or 30 years? So the technician is a very important person in the science. The next category of scientist or science that most of us are familiar with is the so-called inventor. The inventor is a person who essentially makes new devices or methods to ease the process of investigation of scientific problems. So I have here a very famous inventor. I wonder if any of you all know what he invented. Um, is it interactive, or should I just take it as a rhetoric and move on?
They can type in the chat.
Okay, and how do I access the chat?
Oh, I actually have, I mean,
okay, no worries. So anyhow, some of you might know that Zacharias Janssen was the inventor of the microscope. Although not many great discoveries are attributed to him. In fact, very few discoveries are attributed to him. But his invention has led into a great technological feat in that because of it, we are able to analyze microbial life forms or look at microbial life forms. And his own work inspired many such people. This is maybe 1800s, I think. And then you have people like Leeuwenhoek, who looked at a lot of microbial life. But the primary inventor is a very important type of scientist that is out there. Now, when you look at computational biology or bioinformatics, there is no greater example than the inventors that are out there in this slide: Michael Levitt, Martin Karplus, and Varshel. Essentially, they were some of the pioneers in writing computer programs for studying molecular conformations. You may have heard about CHARM and such kinds of tools that are used to build models of protein structures. So you know that you have various ways to study molecules. You may have NMR, you may have electron microscopy, you may have X-ray crystallography and the like. But you need to, at the end of the day, build models out of this. So these were pioneers in building these model tools. And in fact, they are the recipients of the Nobel Prize for Chemistry in, I think, 2013. So they played a very important role in the fact that in that we could convert using their tools that they invented, they actually conceived. You can actually apply or prod through molecular information in the form of models, which is how most of us work with at the end of the day, which is what most of us work with, some model or the other of proteins, of life forms, or of information in general. Now, indeed, in the space of bioinformatics, computational biology, there have been some remarkable tools that have revolutionized our ability to understand information. And one of the key tools that has resulted that I think is central to computational biology, to this whole genomic revolution, is what are the BLAST and SciBLAST search tools of comparative analysis of looking at nucleic acids or nucleotides and proteins. You might have already had a brief overview of what these BLAST searches are, but the point here is that you have to remember that when you're, for example, just think about this, that you understand that you have a large database of protein sequences, and now you wish to find relationships between different proteins. So you have to have a way of querying it and looking at comparing this query with other protein sequences. You may think of doing a word search, but say you hit a certain word, a certain match pattern. How significant is that pattern? And so one of the key things about aspects about the gap blast and SciBLAST methods and tools is the understanding that in reality, that if you have two match sequences, you have to give a score. And so what is the nature of the score? How would you design a score that actually reflects relationships? So one of the things that was done over here was one of the realizations was that the scores are actually not normally distributed, but it shows a distribution known as the extreme value distribution. And this um, revolutionized the way that this tool could be built in making discoveries about protein relationships. Similarly, another tool which is used in computational biology by protein structure, people who study protein structures, is the DALI database for protein structure comparison. Here, the concept is essentially taking the structures of proteins and looking at them in a particular kind of geometrical way by which you can then make comparisons between protein structures and determine if there are related folds between protein structures. So here is another tool, and for example, Lisa Holm and Chris Sander, who invented this tool. There's been no better tool than this, even though this is over now 20, 25 years old, or perhaps even more. Similarly, when the complexity of information improves, and then you realize that there are different ways of grouping sequences as homologous sequences, you can get what is called as a Markov model of these sequences. And then now you can compare Markov models of these sequences to find relationships between sequences, and in fact, the HH spread is a classical example of that, whereby many discoveries that are not necessarily possible by sequence comparisons using BLAST can now be made using this extra sensitive information. But in all this, the key aspect is the development of a tool or the invention of a tool that uses certain concepts. Perhaps some of these concepts are very well known in the sciences, but putting them together in a way that results in discovery of new biology. So invention of tools is a very important aspect of the science of, like in any science, also in bioinformatics or computational biology. Now, the third category of scientists that one can conceive can be called as a symbolist. And a symbolist is essentially a person who creates a kind of symbolic language to describe a science, a set of observations. Often it involves bringing in very rigorous formalism so that you have a certain mathematical basis for that. For example, most of you all, especially those in high school, might know about Erwin Schrödinger, whose wave function was a very critical development in the understanding of quantum mechanical phenomena. Some of you might know about a very famous biologist, in fact, his name is Motu Kimura, who was very important in developing certain mathematical models known as diffusion equations for genetics. And in fact, that the use of this genetics resulted in in the development of what is known as the neutral theory of evolution, which is that most genes are evolving at a neutral rate. There's no particular speed or advantage of all of them evolving fast or slow. I mean, if you look at most genes of an organism, they are evolving in a stochastic and neutral way. And this only came out because of the formalism developed by Motu Kimura in the 60s and 70s. In terms of computational biology or bioinformatics, what is the kind of symbolism that has been developed to make discoveries? And this is a bit harder because I didn't want to bring in any mathematics, but one of the things where you do see a certain kind of symbolism or symbolic logic is in understanding unknown information. So what I mean by this is that you all must have heard of sequence searches. You have a certain sequence, you don't know what it does, and then you say you have this, and then you run sequence searches. You have a sequence, you run it through a database to find homologous sequences, say using SciBlast. Then you get a bunch of sequences, bunch of relationships, and then you say, okay, oh, these look very similar to the sequence that I started with. And then you go on, you make alignments, and then you realize, hey, um, these have certain conserved residues, and you can predict a fold. Sometimes you can be very accurate about your fold because you have structural representatives that are available, or you use structural tools to make these predictions. But the symbolic aspect of it that I wish to emphasize here is when you're trying to make information, when you're trying to make biological inference based on a certain type of information. So now let's say you have an unknown protein sequence, you run through all these processes, and then you have the unknown protein sequence and its homologs in the various genomes of life. What we've realized is that there's a certain way to understand the biology of this protein, and that is generally termed as contextual analysis. So if you look at prokaryotic organisms, if you look at bacteria and archaea, their genes are organized in what are called as operons. In that, what happens is that cluster of genes have a common regulatory mode, which means they're expressed at the same time or they are not expressed at the same time, but they are linked in a particular way so that they are made together. So think about this. So you have gene neighborhoods, you have known genes A and B, and then you have other genes that are associated with it. And when you see these known genes A and B coming together in many, many different organisms, um, one of the realizations that came about when looking at genomic information across, say, bacteria and archaea, is that genes that function together in a common pathway, either say they're making a certain substance, or for example, the genes that are involved in making the ribosomal proteins, which are important for translation, or for that matter, genes that are involved in the biochemical synthesis of a certain compound, or genes that are involved, say, in the glycolysis pathway, all of which you may have learned or at least have a hint of. These genes tend to be clustered in the same locus in the organism. And the advantage of this clustering in prokaryotes is that they're expressed at the same time. So if you have 5 proteins, they're all expressed at the same time, they're made at the same time, so you know that they'll have a greater chance of working more efficiently together. Now, in eukaryotes, which are organisms with nuclei, the concept is somewhat different, and we'll come to that later. But anyhow, the symbolic logic that is important here is that if you find your unknown gene being associated with, say, a gene locus in many different instances, then you know that it is involved in a particular pathway. So not only are gene fusion events, so if you, for example, know what gene B does, but you don't know what gene A does, and you see that gene A is always coming with gene B, so you already have a hint that, hey, gene A is involved with gene B, perhaps in a pathway in which gene B is involved, perhaps it's an enzyme that makes a product, or perhaps these two need to work together to do something, or it's a regulatory pair of genes that are involved in something. So in prokaryotes, you have 2 aspects. This is one kind of logic where genes that function together are in the same operon, or often what happens is that these genes fuse together to make a single protein. So here, these are distinct transcriptional units, but then their mRNAs fuse in a way so that it makes just one protein, which has 2 domains, one which corresponds to the A part and one which corresponds to the B part. So when you see such kinds of gene fusion events, or when you see such kinds of functional physical linkages of genes, then people realize that you can predict that these are likely to function together. And in fact, this has been a bedrock for the discovery of a large number of systems and very interesting biologies. It's a very simple symbolic logic, but very profound in the kinds of things that you can do with it. Okay, so I won't dwell on this more. The next type of scientist is what one can classify as the explorer and collector. And by this, I mean, these are the kinds of people who make foundational observations, or they're not just, for example, Linnaeus was looking at things that are already there, but foundational observations involve new frontiers of data, and they are on the lookout for interesting phenomena. And one of the great examples of this is the astronomer Tycho Brahe, who studied the motions of many planets. And because of his accurate observations of and collection of data, we are able to understand the motion of planets. In fact, many interesting scientific principles and theories came out only because we had observations like Tycho Brahe. Indeed, there are many explorers and collectors that are there in biology. And in fact, in computational biology, this is something that is really fun to do. So back to the same problem, you have a certain sequence, and how do you find out you have a certain protein sequence as shown in this? How do we get biology out of this? How do I know what this is actually doing? Now, again, using the same methods that I just spoke about, you can make, you can reach a certain level of discovery. And sometimes just by what you call very systematic analysis and looking out for such kinds of data, you're likely to make some very profound discoveries. So let me describe a certain discovery that we made in our lab. This was 20 years, not 20, maybe about 15 years back. Now, when the sequences of various organisms were made available, one organism that was very highly prized in that sequencing in the time, and lots of interesting organisms were sequenced, were those that are called as the apicomplexans. Now, these are organisms such as Plasmodium falciparum that causes malaria. These are often parasites of animals. Toxoplasma, for example, causes it's basically transmitted through the gut, but it can also go into the brain and cause certain behavioral changes. Then there are those such as Cryptosporidium. These are apicomplexans that that cause a diarrhea, and they have a gut-mouth cycle. So a lot of these organisms are related. They were called apicomplexans because they have a very interesting microscopic feature. They have a certain alga that they have swallowed in the distant past called as the apicoplast. And so when I made the slide, these were the organisms that were that were sequenced then. But when they were sequenced, so here are the kinds of things they do. You have malaria, then you have theileria, which essentially causes a lymphoma. It transforms your white blood cells and you get a kind of cancer with it. Or there's Cryptosporidium that replicates in the human gut and causes a kind of dysentery or diarrhea. So when these genomes were sequenced, one of the big mysteries was that what are the transcription factors? And I'm guessing that most of you all have a general idea of what transcription factors are. Very briefly, these are proteins that are involved in regulating the transcription of a gene. So you have the basic transcription apparatus that makes mRNA, but then there are these other proteins that determine when a particular mRNA needs to be made. So, we had the model organisms such as Drosophila and humans, plants, and even yeast to look up to find similar transcription factors. So when we looked at this, the initial pass, when people sequenced this genome, one of the surprising things that struck them was that there are no transcription factors. And in fact, people started theorizing that, hey, maybe this is an organism that does not require what we call as a classical transcription factor. And it might there might be another way of regulating its transcriptional program. But here is where the explorer and collector is very important. Now, when you very carefully ply through the genome and look for certain DNA binding domains that are likely to be transcription factors, you can, for example, make a discovery. So in our group, we then discovered that in fact, in these epicomplexins, there's a very particular type of DNA binding domain that is expanded in them. And it belongs to a certain fold called as the AP2 fold, or the AP2 superfamily of proteins. And the AP2s are actually are studied in transposons, where the DNA binding part of the transposon has this AP2 domain. Also, in many plants, there are many transcription factors that have this fold. So it then, and this unification is not very trivial to make at the sequence level. It's not like I just run one blast search and say, oh, I got this transcription factor that is there in plants and epicomplexes. It in fact involved mining through the genome, being very observant of what the hits, sequence search hits are before we could arrive at this. And based on this, of course, once you get it, then you can do the rest of it. That is, you can align these sequences, find out what the conserved residues are, and also get predicted secondary structure, and and so on. And in fact, this discovery led to a major revolution in epicomplexin biology, because now you had the 20 or 30 transcription factors that you need to understand how genes are regulated in these epicomplexins at different points of time in their lives. As some of you all might have studied the malaria cycle, you have something in the mosquito, you have something in the gut, you have something in the liver, and so on. So, so at these different stages, you have different genes that need to be turned on. And now with this framework, with this discovery, people actually were now able to study these particular genes and see how they are first made in the different stages of the life cycle of the parasite, or contribute to the expression of or regulation of expression of different genes at the different stages of the life cycle of the parasite. And in fact, this is now a bedrock of epicomplexin biology, but the basis of this was entirely computational. Okay, so sort of zooming out, if you now, from this emerged a very general principle, and this slide just shows that is, if you start looking at transcription factors in this way, that is looking for DNA binding domains, you notice a very interesting thing, that there are certain families that tend to explode in different, or explode in the sense that they are present in large numbers or are overrepresented in certain organisms. So here is essentially a table, if you want, these are tables where the blue shows that a gene or a transcription factor is present, and white means it is absent. These show that these are present in a very high, or these are, they show a very large number of transcription factors in each genome of this organism. And now the backdrop, this is the evolutionary tree of eukaryotes. And I wouldn't want to belabor you all too much with that, but this is a very important topic in computational biology, is to try and understand what is the relationship between these different organisms. How are they related? For example, we now know that animals and fungi are actually closely related, and outside them are what are called as amoebozoans, such as Clostridium and entamoeba. And outside that are the other organisms. So different groups of organisms can be grouped together. For example, the apicomplexans that I spoke to you about are actually very closely related to ciliates. Many of you may know of Paramecium and Tetrahymena. But ciliates don't have this expansion of AP2 that apicomplexans have. So what it shows is that different organisms or in different lineages of life, different proteins become the primary transcription factor in that organism. So for example, AP2, it's absent in many eukaryotic lineages, but it is greatly expanded, for example, in plants and also in apicomplexans. Another DNA binding domain called VP1 is only there in plants and in some relatives of the plants, chlorophyte alga. Animals have an entirely different profile of transcription factors that are expanded. So this type of expansion is known as an LSC, or a lineage-specific expansion. What it means is that this gene family is greatly expanded in that particular genome. So the general principle is that so if transcription factors are so important for regulating genes, then how come they change so easily? So what seems to be the general biological observation that comes out of this exploration is that although all the genes that are regulated by these are shared between many organisms, the regulators of these genes can very easily change. So to give you an analogy, it's very easy for a regime change to occur in the eukaryotes. An entire regulatory setup can be completely taken over by a new set of proteins, which I think is a very interesting observation that can only come when you start looking at information at such a level with such depth. It is not very obvious when you are just looking at them in a lab, or when you're studying one organism, or when you didn't have this kind of information. Okay, so this is the basic point, that is, as a result of independent lineage-specific expansions, the most utilized transcription factor differs between the different lineages. Okay, so now the next kind of scientist is what you may call the ontologist. The ontologist is a person who tries to find order in all this data that is accumulated. So he defines various categories into which you can classify these observations. And he brings out relationships between different categories and makes predictions based on these. So for example, one of the greatest examples of this is the periodic table of Dmitry Mendeleev, who essentially, using this ontological scheme, developed and using concepts such as atomic weight and atomic number, was able to classify the elements in an extraordinarily logical way that actually revolutionized our understanding of chemistry. So what is the role of ontology in, say, computational biology? In fact, it is a very central role in computational biology. Only after we began to get information from all these genomes were we able to put it all together and ask very systematic, deep questions. So for example, all life can be broken into three major branches: the bacteria, the archaea, and the eukaryotes. And this only came, if you look at a bacteria and an archaeum, you may say, hey, what's the difference between? They all look like little rods and spheres. But in reality, when you start looking at the genetic information in them, say, for example, you look at the kinds of ribosomal proteins, or you look at the proteins that are involved in replication and transcription, they're very different between these two prototypes of organisms. And then came the big realization that was first proposed very early on, before all this information was there, and was rigorously shown by computational analysis, that in fact the eukaryotes, the organisms with a nucleus, are in fact a fusion between an archaea and a bacterium. And in order, we are always trying to look at when you look at different pathways that are very specific to eukaryotes, for example, how are proteins transported from inside and outside the nucleus, or how are genes regulated in eukaryotes, you find that it has components that have both bacterial origin and archaeal origin, and they have come together in a very interesting way to give rise to the eukaryote. So, in a sense, this ontological information this ontology is only possible when you have this kind of information. But let me show you something that which might be somewhat related to what you have learned over the past week. Now, here was a discovery again made in our group a while back. The first thing was that there are these proteins. So most of you may know of histones. Histones are proteins around which DNA is wrapped in eukaryotes and archaea. Now, histones actually get modified. They get modified by a methyl group by a certain set of proteins. And early on, there were these very interesting proteins that people found, and they found these in proteins that go into the nucleus and interact with chromatin, and they didn't know what they did. So early on, work from our group using the same techniques of comparing protein sequences and structures, we arrived at the realization that in reality, these proteins are enzymes, and we could even predict what type of an enzyme it is in that. So for example, let's say this is a side chain, this is a lysine side chain of a histone, and so here it is, and here's the methyl group in the lysine of side chain of histone. Now, what happens is that as a result of the activity of this enzyme, this methyl group gets hydroxylated, and then this hydroxymethylene exits as formaldehyde, and now you have a demethylated amino acid residue. So the methylation is done by a certain set of proteins. Here you can see that these, for example, in black, are all the proteins that methylate the histone. And these in the red are those that demethylate the histones. So in fact, maybe you can go and read up. There's something known as the histone code. So in eukaryotes, I showed you that in prokaryotes and bacteria and archaea, you have operons that regulate genes. But in eukaryotes, the structure of the nucleus or the nuclear DNA is quite complex. And the regulation of that is mediated by a vast array of proteins that are perhaps of bacterial or archaeal origin, but they're not used in the same context in bacteria and archaea. So for example, I just showed you that this chemistry that, these proteins that were called as the Jod proteins were predicted to cause this. And over time, it was confirmed experimentally that indeed these are all different types of Jod proteins from here to here. They are involved in different types of demethylations in different parts of the sequences of histone. So for example, here's histone H3. You can see that these are the lysines or sometimes arginines in which these methyl modifications are made. And these are the lysines where the methyl group is removed. So these are the enzymes that catalyze that. Prediction was first made entirely computationally. But the point here is that now that you have this set of proteins, the next big question that one can ask is, where do they come from, right? Were they there right from the beginning of life, or are they derived from other proteins? So there are different scales at which you can analyze such kind of data. The first type of scale is to first make a phylogenetic tree. So if you make a phylogenetic tree, so these are all these proteins that have that enzymatic domain, and you can first find out what the relationships between these different protein families are. That is at one level. Then you can, at another level, you can see which organisms are they present. Now, for example, if something is there in all eukaryotes, then you know that, hey, this must have been in the last last ancestor of last common ancestor of the eukaryotes. But you know, bacteria. So one of the things that is obvious in this is that, say, if you think of the evolution of life, say life evolved 3 billion years ago, and then the archaea and bacteria came. They should have come first because the eukaryotes are a fusion of one type of bacterium and one type of archaeon. And now we in fact know which type of bacterium was fused into making the eukaryotes and which possible type of archaeon went into this making of this. So one obvious thing is that the bacteria and the archaea had already diversified into many different types before eukaryotes were formed. So you can now make further inferences that, hey, these were perhaps present in the last eukaryotic common ancestor, and these families, very few of them, were perhaps present much earlier. So you can then, using structure comparisons, so if you know that what type of structure does this belong to, you can ask the question, are there related protein structures? And if you go deeper, you can group these with respect to other protein structures. So using tools such as DALI, these families of enzymes are classified as, were shown to be related to cupins, which are other types of sugar binding domains. And you can see that whereas this is an enzyme, many of these are not necessarily enzymes. And in fact, in the same set of folds come what are known as the viral capsids. In fact, the most interesting thing about viruses is that although there are many different types of viruses, the protein that makes the capsid of the virus is of the same fold across many different viral groups. So now you can group these different proteins together in a structural viewpoint and then do the same type of analysis that you did for these proteins across all these other families, and then ask, okay, which of them would have been there, say, in the last eukaryotic common ancestor? Which of them were there in the last bacterial common ancestor, last archaeal common ancestor? Now, if something was there in the last common ancestor of all life, which of them can be traced to the last common ancestor of all life? So this type of information is known as ontological information. And this is extremely useful in trying to make sense of what a particular unknown protein is doing. Because once you have such a scheme, tomorrow a protein, you find a protein, say you crystallize a protein and you find, hey, it belongs to this group. You can now use this ontological information to derive what it might be possibly doing based on its relationships to these different families. So here you can see that there are so many different families that I spoke to you about. So here is the enzyme that which we predicted would cause this reaction, but there are many other domains that all share the same core that you see in green and blue. And whereas the core is the green part is the core, there are other parts that are unique to these different families. Just like, comparative paleontology, you have a basic framework of the bones that describe a vertebrate, but then different bones are proportioned in different ways across different organisms. And based on that, you can make conclusions about relationships between them. So this ontology can grow into a very general theory. So one of the big questions that has often been posed and that is often repeatedly posed is how many folds are there? So if proteins can all be grouped together and relationships can be found between each other, how many different protein folds are there in this universe? Is it limited or unlimited? So when you ask these questions, one of the first things that strikes you is that you need information for protein folds across all life forms. And now we have that because we have over 10,000 genomes from very distinct life groups, you can now ask these questions as to, okay, the biggest realization is that 95% proteins or more can be grouped into about, maybe 8 to 10,000 folds, which is a very small number of folds in comparison to the number of genes an organism, or the number of genes that we have sequence information about. So much of the protein universe can actually be contracted into a small set of related protein folds that have gone to give rise to life, which I think is a very profound insight that comes only because of this ontological classification. And then you can now go on to make theories as to how for example, all proteins which just have alpha helices in them, all protein folds that have alpha helices in them, so how they may have evolved um, based on very simple frameworks such as duplication or discontinuity causing folding. So from a simple alpha helix, you can get superhelical repeats, you can get complexly folded protein domains, or small parts that are not. So ontology is a very important aspect of understanding information. So, and the ontologist has a very key role in this. And finally, if I don't have, I think I've overshot my time, and I hope it's not, I'm allowed a few more minutes. I'll finish very soon.
Yes, go ahead.
Yeah, finally, at the end of all this, the sixth category of person, the last category, is the conceptualist. The conceptualist is essentially a person who takes all this information and produces new theories or discovers a new law that explains order of some kind, discerned by, say, the ontologists or the explorer, all these other categories that we saw earlier. One of the classic examples of this is Charles Darwin. Now, it isn't that the evolutionary theory wasn't sort of gleaned before Darwin. In fact, many people had a clear idea that organisms evolved, that there's an evolutionary scheme by which organisms evolve. But Charles Darwin was the first one to actually propose a theoretical basis, that is a basis, which is natural selection, as being one of the fundamental basis of evolution of life. So of course, he not just natural selection, he actually looks at various nuances of what this whole natural selection means and so on. And this only came as a result of synthesizing information about a vast amount of life forms. And this type of conceptual leaps can also be made in computational biology. And in fact, one realm in which I shall describe this, and there are many, thousands of such discoveries of concepts that have origins in computational analysis. One thing that I shall talk about right now is comes from our group, where we made interesting discoveries on biological conflict systems. Now, you know that much of life is actually in some form of conflict or the other. So this can be between organisms of different types, organisms of the same type, between that is interspecies, intraspecies, and also within a genome. You have a genome that has many genes, and these are also sometimes often in competition with each other to make sure that they are propagated into the next generation. So with this large amount of information, we now have this great opportunity to actually study evolution in action. And as I said in the beginning, one of the great advantages of having a large amount of information is that you're actually, the evolutionary experiments have already been done. And it is for us to understand this grammar of evolution to make sense, to make biological discoveries. So very briefly, we were studying some proteins, and one type of proteins caught our attention. And these were very huge proteins. There was few things that were known. They were secreted proteins, and they have these repeats that suggest that they were present as filaments. But at the end of each of these was a very interesting enzymatic domain. Often these are nucleases that cut DNA. They could modify DNA. Some of them modified proteins. Some of them modified. Some of them, were pore forming. So essentially, all of these can generally be classified or categorized as toxins. So across different life forms, prokaryotic life forms, we notice this pattern. So at the very end terminus, you have domains, protein domains that are involved in transport of the protein through a particular pathway. Then there are these filamentous repeats that actually increase the length of the protein. And at the end are these toxin domains. Now, when you start looking at this more carefully, you realize that when you look at the genes of these domains, so these are, for example, this is in proteobacteria, these are in actinobacteria, and all other different types of bacteria. So the pattern is very similar. There are the N-terminal domains involved in secretion, then there are these middle domains, and then you have these C-terminal toxins. Now, if you look at the gene locus of this, in fact, they have a very interesting gene organization. So this is the gene that makes a toxin. And next to it is a gene that we predicted would be the immunity protein against this toxin. And in fact, this was shown to be so. And after this were many genes that had little cassettes of very different types of toxins and immunity proteins. So this is the primary locus, and then you have these cassettes of toxin and immunity, and so on. So based on this, what we realized was that you are getting a toxin immunity pair in the neighborhood. These are cassettes, and this is the primary locus. So there is some way by which the organisms decide to switch a particular toxin and immunity pair at different points of time. And in fact, what we saw was that if you look at very similar organisms, say 2 strains of Escherichia coli or 2 strains of Bacillus subtilis, they would be identical in all aspects up until this point of the toxin and immunity protein. And often this pair would be in the neighborhood and something else would have come in. So there was some kind of switching going on in the different organisms. And one could sort of postulate that these cassettes actually are put in and they are removed, and a new cassette comes in, and then you have a new set of toxin and immunity pairs. But what is the biological context of this? And why is it that closely related organisms are constantly changing this? Why is there this constant selective pressure for diversification? And in a sense, it resembles, if you all know how antibody genes are assembled, it sort of resembles that process. So when you start looking at this more carefully, you start looking at the toxins as to where they are all present. One of the things you notice is that many of these toxins are present in what are called as coalescents that are involved in lysing. They are made by plasmids, and these are addictive modules. That is, if a host cell wants to kick out a plasmid, then so these are plasmids that have these toxin and immunity protein. Now, if a host kicks out this plasmid, the toxin is there and the immunity is not made, and this host cell dies. So it's a kind of addiction module that makes sure that that the host maintains a plasmid. So this was around, but then the conceptual leap that we made was that perhaps this is a way whereby organisms are able to distinguish between self and non-self. Now, in terms of larger macroscopic organisms such as ourselves, we easily use our sense organs to distinguish those that are like us and not like us. But what about bacteria? And then came this remarkable conceptual advance based on this. That is, this is actually a way whereby organisms talk to each other. Organisms talk to each other and decide if they are of the same kin or not. So, for example, let's take this case. So you remember that the proteins had very long repeats, and at the end they have this toxin tip, and they are secreted. So they're on the surface of this hypothetical bacterium. Now, this is another bacterium that is related to this. Now, the theory then was that this toxin is injected into the relative or is speared into the relative. Now, for this toxin, there's an immunity protein that I showed you was there in the locus. If these two are related, they will have the same immunity protein. So if this toxin gets into the cell, and it will be blocked by the same immunity protein. But say if this toxin gets into another cell, an unrelated cell, then since there is no immunity against this toxin, it will kill the cell. So this was a way whereby organisms were able to recognize each other and cause, and hence you get colonial cooperation. And that's how organisms are able to group together in certain environments. So now there were many themes that we predicted just entirely based on the initial observation. That is, you'll have toxins that are like spears and others that are like bullets, because they're much shorter toxins. They'll be shot out into the medium. Organisms that are similar will survive because they have this immunity protein. Organisms that are different won't have the immunity protein and will be killed. Now, in all this, of course, when you start looking at biological information, it's not always black and white. So now you realize that there are many. So if you, for example, take organisms that stay in your oral cavity, there are, for most of you who might know this, there are at least 200 species of bacteria that are there in anybody's mouth at any given point of time. And these 200 species of bacteria are, in fact, the interesting thing is no two people have the same type or same ratio of different bacterial types. So they're jostling for resources in your mouth. And when you look at organisms that are, say, just there in their mouth, you find these toxins and immunity proteins are very are present in all of them. But then there are some others who are what you may call as cheaters. Then they have a large number of immunity proteins. They don't have toxins. So if you throw various toxins at them, they're going to be able to neutralize all of them. And they will be able to grow along with an unrelated kin. So in a sense, trying to cheat and get resources from the other. So this biological theory came out of just by looking at protein sequences. And hence, this is what an example of a conceptual leap that came out entirely from computation analysis. And in fact, this has been experimentally confirmed. There's a large field of biology that has come as a result of this basic hypothesis and discovery. And people have found cheaters and whatnot. All right. So I would like to end. So having spoken about the different kinds of scientists that are out there, some of you have a feel. There is no one particular type of scientist who's greater than the other. You need all of them to bake this cake and understand life. But I would like to leave you all with a certain few impressions that are interesting. So one of the big questions that I often had as a high school student was that how many species are out there? If we were able to count all the species, what would be the number? Many very smart people have tried to look at this based on sample, based on getting various samples, and there are various counts. So if you assume that one such as, say, by this group, this paper said that there are 10 million species on Earth, and out of which only 11,000 are prokaryotic. And what they said was that the 86% of land species and 91% of marine species are yet to be discovered. And if we go at this rate, it would take us about 480 years to comprehensively catalog Earth's life forms. And this was done about 5 years back. So we still are far from cataloging all of life. A typical professional taxonomist classifies about 25 species in his or her lifetime. This is a very low bound. In fact, what seems to be coming out is that there are between 10 to 100 million species. And what is most striking and interesting is that most life is microscopic. So what life you see around you is not really 90, is basically 5% of life. We are just large, that's all. We don't see small life, not just bacteria and archaea that are invisible. There are a large number of eukaryotes that are also invisible to the eye. Their own biology is not simple. It's also extremely complex. It's also extremely diverse. And we are just beginning to scratch the surface of this information based on where we stand today. So a couple of things. If you have, I mean, I am greatly inspired by E.O. Wilson. Many of you know that he's one of the most famous ecologists who's made many fundamental contributions to biology. And E.O. Wilson said in one of his books that if I were to do it all over again, I would be a microbial ecologist, because 10 billion bacteria living in a gram of ordinary soil has so much biology, none of which is known to science. So he goes on to say many things, of which he says, I would cut my way through clonal forests, sprawled across grains of sand, travel in an imagined submarine through drops of water, proportionally the size of lakes, and track predators and prey in order to discover new lifeways and alien footprints. So we are barely at the beginning of this process, and there's so much to discover. In fact, just if we were to look at our own relatives, and many of these pictures are taken with a paper microscope. Show you all the paper that I have at home. In fact, all these are paper microscopes. This is, for example, a tardigrade. This is an animal that can survive under very extreme heat and cold. We know very little about this. We know some things, but very little. There are rotifers that are microscopic, and everywhere you just walk 5 meters from your house, take some soil, or take a sample of water, you're likely to find a rotifer. One of the interesting things about rotifers is there are some species of rotifers where no sex has been observed. So then how do they overcome mutations is a very big question in biology. But we do not have enough people doing these experiments. But yet there are these questions that are likely, possibly answerable entirely using computational approaches. So I want to leave you all with this video I took with the same paper microscope. Look at this organism here. This is from a pond very close to where I live. And they kind of go around this way. This is a flagellate you can make out. And then they talk to each other, and then they talk for some time. There's some kind of interaction between these organisms. And then they just fly away, or they just sort of swim away. So I know that this is a flagellate, but this hasn't been classified in biology. I really don't know what this is. Nor do I know what is happening over here, what genes are expressed, what profiles of transcription or protein interactions occur in this process. So I gave it a name, synchronous disaggregation in a mysterious flagellate. But here is the challenge. Here's another challenge, a computational challenge. You look at something like this. How do I know what this is? So one of the big revolutions that we are likely to see in the coming years is imaging, computational imaging. To be able to classify images, to be able to just look at videos and images such as this and extract information about what it is. I think it is still a big challenge that remains to be solved. Anyhow, I think I've spoken much, and I'll be happy to take any questions if I have time. But thank you, and for those of you all who are in school, keep learning.
Thank you, Lax. That was inspiring, and I'm sure that there will be lots of discoveries, correct? Like Lax actually has walked you through step by step of how to do discoveries. He has made a lot of discoveries, and I'm sure it was inspiring for all of you. So I'm just going to give a minute to see if any of you have questions. I think one question that from Karina was, Lax, which category would you classify yourself under?
Oh,
I
well,
guess
I can only perhaps explorer, explorer, collector, and perhaps one who's made some fundamental contributions to concepts.
Inventor.
Yeah. So as I said, there's no one particular category, but I'm not an inventor in the sense I didn't develop any tools, but more of an explorer of information and also developing concepts, developing new biology based on this exploration. So yeah, maybe 2 categories, 2 of 6. That's all I can claim.
Any questions? Do any of you have any questions? It doesn't have to be a biological question, but do you have anything else? Any questions from anyone? You don't have to be shy. Everybody is in wow and in a state of shock, I'm sure, right?
Or perhaps totally confused at the end of this.
Anyway, so I think I also Lax actually introduced you to an outstanding tool that I would like all of you to actually go look at. It's called Folderscope. It's just a paper microscope, but it's an extremely high resolution microscope. And you can actually just explore, go to a pond or, just take something and you can actually look at these organisms and very interesting things. So there is one question from Megan. What motivates you in your occupation?
That there's a lot to discover out there. That in fact, biology is not limited. It's sort of limitless. And being a theoretician gives you a handle to study many distinct kinds of problems. Normally in an experimental lab, you're focused on one organism, one small problem, but in theoretical biology, you can actually study a vast range of life. And that gives you a much deeper picture of life than, that's what I feel. And that's what motivated me. But hey, for example, there's something interesting even in the SARS viruses that haven't been discovered yet that will motivate me to look at it. So that's the kind of thing that there's a lot to be discovered. That's what motivates me.
And there is several questions actually. So how did you, I'm going to read all those questions. So maybe if you are able to.
I do see some of them.
So you're able to see, okay.
Yeah.
So
how did you start your work with NCBI?
How do you confront?
Okay, so I was an experimentalist, and as I told you, when these genomes were coming out, I was pretty disillusioned that all these other people are having access to this information. They have all these nice computers and tools, whereas I'm working on this plant virus, and maybe I will make contributions to virology, but I wanted to have a much broader view. So I was lucky that in those days it was fairly less competitive uh, to switch fields from an experimentalist to a theoretician. I also had made some studies of my own, and that helped me, get a look in. And I have not looked back ever since. How do you confront challenges or setbacks in your career? Scientific careers are long; they can be depressing. All of us have go through it. I think there's no other way but to keep at it, to be positive, and to actually be real about what you can do, what you cannot do. You're very idealistic when you're young, but when you start doing stuff, you begin to realize the limits, your own limits. But at the same time, you realize where there is a niche for you. One of the things in science that I learned was that you shouldn't be doing what is hot and what everybody is doing, because you would be just one amongst the many. You should look at fields and subjects and problems that very few people have studied. So one of the key aspects of being a scientist and being happy with your science is to find those problems that are interesting. And challenges often are more of a personal nature rather than scientific nature in a scientist with a long career. So, you if you're married, you have a family, you have to take care of them, or, you can't go to work under such circumstances like coronavirus pandemic that we have here. So you just have to stay positive, and as long as there are interesting problems that you can think about and address, you keep at it. I think that's how I face mine. Yeah, my background and research interest. Yeah, so my background, My undergraduate was in microbiology. I particularly like chemistry and physics, but I chose to do microbiology. And I then did a master's in biotechnology, all this in India. I came here for my PhD in plant virology. In those days, there was a very big problem that, is if you, that is transforming plant itself was a big issue. Can you make transgenic plants? Now, nobody likes GMOs, but when I did my work, just getting a GMO plant was a very big deal. And then people realized that when you make a GMO plant, the protein that you make which is genetically in the genetically modified plant, is not expressed often. So what is that? And that actually led to a whole field called as RNAi. Um, and I was there in the early parts of that process. So then after that, my research interests are, one thing I realized was that I'm more of a person who likes broad views. I like to read a lot of textbooks. I'm one of those guys who likes to read many different topics in textbooks and feel very happy that I know many different things. But a scientist need not actually be that way. Not all scientists have to know everything. In fact, some of the great scientists are very limited. They know only about their organism. They may not have a general idea of even biological theory, but they do make some very profound discoveries in what they do. So research interests are broad and they're constantly changing based on this. So what is your most favorite or interesting discovery and why? There are no favorites. I only study a problem if I feel that it's of interest to me. I may not take up a problem if I find it's boring. But I can give you some examples. Like, for example, for the longest period in the history of biology, people knew that DNA was methylated. And at some stage, the DNA, the methyl group was removed from the DNA. But nobody knew how this would happen. And it was one of the big black boxes, big mysteries. So we actually arrived at this using a very, there's this other organism, a trypanosome, which modifies its DNA. And then we found a protein domain that is there and that is also there in humans. So based on that, we predicted that actually humans will have a very distinct modification called hydroxymethyl cytosine. And this was entirely done computationally. Luckily, we had a very good collaborator, and it was shown to be so. It has, it's shown to be relevant in cancer and epigenetics, behavior, and whatnot. So I think I would rate that as one of my most interesting and favorite discoveries, that we could actually predict a DNA modification entirely from theoretical principles. But in terms of what I really feel very great about is that, most of y'all may know that RNA polymerases were that make transcripts. When the RNA polymerase was crystallized, they said it looks like nothing. It looks like something very different and distinct. But in fact, using ontological tools and computational tools and very careful analysis, we were able to dissect this most fundamental aspect of molecule of life. And the inferences that I understood that, life is actually the complexity of life, that there was a simpler time in life, and then there was assembly of complexity based on assembly of various domains. I think that was very profound. Have you ever started a project that you thought was interesting? Yeah, of course. There are many projects that, for any scientist, it often happens that you start with something that is interesting. You might have a hypothesis, but you might be proved wrong. But what often happens in a scientist's life is that somebody else does it before you, or somebody has already done it 15 years back, and you were lazy and you did not look at, you did not go back and look at the literature. You always keep looking. As a theoretician, one advantage is you always keep looking for new kinds of things. Are there limitations in the amount of funding one can receive? Yes, it is one of the most limiting things for any researcher. You have no money, you can't do research. But, in fact, as a researcher, I even feel fortunate that I'm able to pursue what I'm doing, this kinds of pursuits are not commonly done by people. You don't have the time to sit down and sit in front of a computer and look at stuff and make discoveries. But I think it's great that, say, for example, in my case, the United States government, or also certain universities and organisms are spending this amount of wealth in just exploring information, just trying to find new and interesting things that may have no relevance or at that time, but maybe sometime later it is important. There are limits on the amount of funding you can receive. Yeah, definitely.
Well, Lax is lucky. I can talk to you about lack of funding because I'm in a private institution. So Lax is lucky because he's at NIH. So some of us are struggling without funding. It's a big part of big part. It's a big part of our lives. Yeah,
it's a big part of science, and can be very depressing in general. But the scientific problem often is usually overcomes all that. How do I know if the idea I'm pondering has already been worked on by some other lab, though there's not much anything published by them? Should I go further with that project? You don't. If you are, there's one thing that a scientist. Why do we approach scientific problems is a big question. Not for any fame or to write that paper, but just because it's interesting. So you approach a problem because it's interesting, and if you think you have a solution, great. But if somebody else does it before you, too bad. That's how you should look at it. You should do it for as long as you can, and usually you get a scent of somebody doing it because the scientific community is not that huge. Can you provide some insights for a student from India who's wishing to pursue an MS, PhD in the US? I don't know. The system has changed very greatly. In my days, we had to take certain exams, GRE, and so on. Now they're getting rid of the GRE, is what I heard. If there's any advice I would give a student wanting to do a PhD or an MS in the US, one thing is try to get some research experience, work in an academic lab or in the industry, and that will help you in your case, make your case, and take these exams or whatever, and do well in your studies.
Anyway, that was just wonderful and amazing questions from all of you. I know that we can sit here with Lax for another hours because there's so much to talk about. But thank you, Lax. I know this is, taking time out of your very busy schedule. I join everybody here on the call, thanking you for your time. And as you can see from the response, it's motivated them, and I hope you will see discoveries from them as well. So it's a very different way of thinking. And thank you so much for your time, Lex. Thank you.
Thank you so much. Thank you so much, Sona. Thank you, everybody. And stay safe.
Yeah, I'll talk to you soon. Yeah.
Okay. I can leave now, right?
Yes, yes.
Okay. Thank you all. Bye-bye.
Yeah. Thank you. Bye, Lex.
Thank you so much.
Yeah.
Yeah, thanks.
So, so we had the next, I guess, 30 minutes or so. I think we will continue with what we had, but also I'm going to just walk you through just one resource that we did not do yesterday, which you may want very quickly, about like 5 minutes or so I'll spend on that. And then just discuss an activity that I have put together for you so that you can spend that time on the weekend, right? And then we will kind of, since we don't have much time, we'll kind of summarize what we did. I don't think we will have time for a game. I actually put together a game that we could have played, Jeopardy, but I guess we will not have time for that. But that's hopefully for another time. So let me actually walk you through the resource that I actually did not walk you through yesterday. Okay, guys, so give me 1 second. So this is actually a very important, interesting resource called SCOP, Clusters as Structural Classifications of Proteins. I'm not sure how many of you have actually heard. Alexei Murzin is the person who put together this. And this is another one of my favorite tools. And in fact, Lax and his group actually They use this for discoveries and all that to figure out structural similarities. We have talked this whole week about how two sequences that can have a very low sequence identity, but when you look at their structures, they could still be identical or similar. So how do you classify structures? So in the protein space, how do you classify protein sequences? You classify protein sequences based on their sequence identities, right? You do an alignment, you look at all the sequences that are similar to each other, and then you place them in one family, and relatedness, this is completely based on sequences. But in structural space, you can do BLAST, you can do alignments of sequences because you're not going to be able to detect similarities between two things. Actually, Lax and his group, there's another name I should put together, is Aravind is one of the child prodigies in sequence space. He's also at NCBI, and I've actually worked with Aravind very young. Lax works with Aravind as well. He is also a pioneer. I mean, you all can look up all the papers published from Aravind, Aravind, and Lax as well. And you will see so many things that they have found just looking at structures and relatedness. Of course, it's not easy to duplicate what they're doing, right? You have to have the knowledge base. But here, when you look at structures, just imagine structures in front of your eyes, right? How can you classify structures based on their secondary structural elements? What do you think? How can we classify structures based on helices, strands? That's all we have, right? In structure space, that's all we have. So how can you classify structures based on just these two secondary structural elements? Any ideas how we can classify them? Any ideas that come to your mind? No? Amount, yeah, so actually, okay, yeah, amount of helices. So I would just rephrase, Karina, what we'll say is, we will classify all the structures that have only alpha helices as all alpha proteins based on shapes and structure. That's exactly true. But we'll actually classify them based on their content, right? Secondary structural elements. So we have found there are 4 classifications. You can have all structures that are only alpha helices, all alpha, all structures that have only beta strands, arrangements, that's correct, Misha. So all structures that have only beta strands, which is called all beta. And there are structures that have a mixture of alpha and beta, right? In that classification, you can actually classify them into what is called alpha slash beta or alpha plus beta, which means alpha slash means these are structures that have alpha and beta, but they are arranged in a very specific pattern. I think I showed you those patterns, correct? I showed you structures where they have alpha, beta, so there is a specific structural motif in them. So there is a formula. So after alpha, one comes only beta. After beta, then alpha. So there is a pattern to it in 3-dimensional space. These are proteins that are classified into what is called alpha slash beta proteins. And then there are proteins that have alpha helices and beta strands, but they can be arranged in any different, there's no pattern to it. You know? They're not arranged in any specific way. So these are all called alpha plus beta. So there are 4 major classifications in structure space: alpha, all beta, alpha plus beta, alpha slash. So that is pretty much how most structures are classified. And again, Alexei Murzin, who's come up with this amazing person, brilliant mind, he can just look at a structure and he can really figure out which family it belongs to, which superfamily it belongs to, which, you know, all of those things. Now let's actually try to see which family that our hemoglobin actually belongs to, right? The whole week we have talked about only hemoglobin, right? So what is that we have talked about? So let's 2HBS, you just put in the keyword. If you all want to follow, you can follow along. I'll just give it a second. I think I should actually send you the link. Right? So if you actually go to this place and if you click, so you have the hemoglobin is actually placed into, this is the alpha chain, this is the beta chain. They're placed into different families, right? So if you click on this, for example, it gives you the structure here. What is the domain of this alpha chain? And then if you click here, that says show ancestry, right? So then you have that it belongs to all alpha proteins. Hemoglobin has only alpha helices. It belongs to this family of all alpha helices, class, that's called class. And it belongs to globin-like fold. And then it belongs to the superfamily of globins, which means there are lots of different families. I think I've showed you that hemoglobin has lots of different families. So it belongs to the superfamily of globin. So do you all, what does it mean for proteins that belong to globin-like? What do you think is common among all of the members of this superfamily, which is globin-like? How do you and why do you put things in a superfamily? What do you think is a superfamily? Superfamily means that all these members of this globin-like superfamily actually have the same common ancestor, which means they all evolved from one fold and everything else looks like them. So this is also all of them that have something similar actually belong to the superfamily of globin, which means they all evolved from a universal common ancestor. That's what superfamily belongs. Then within the superfamily, there are members of this that are more closely related in their functions than others. So then you have the family. So hemoglobin belongs to the superfamily of globin-like, it belongs to the fold of globin-like, and it belongs to all alpha proteins, and it has this particular domain. So that is a class, this is a structural classification of proteins that actually you can trace back at the evolution of every single one of them, right? So you can actually go and look at any protein that you want to look at that has a structure. You can just put the PDB ID or UniProt ID here, and then it will tell you which family, superfamily, and fold anything that belongs to. Okay, so this is something that I did not have time to cover yesterday. So I just thought I would cover that, right? Now, before we get to the activity that I have for you, so let's, oh, I see. Okay, screen sharing is wrong. So now what I want to do is just actually, we won't have time to play the game, but at least I'm going to just summarize for you, just to look back at, oh, sorry. 1 second. Okay, can you all see my screen? So, you know, one whole week, it's amazing how quickly time flies, right? It's just amazing how quickly time flies. I can't even believe that I've been with you for this whole week. I hope it was, at least you learned something from it. So this was a slide where we started, right? Day one, introduction, and all of those things, right? So here, of course, this is what we learned in the first lecture, right? We learned about definition of bioinformatics, all the key players in bioinformatics, all the sequence explosion and all the challenges we are facing today, an era of big data. What are the solutions to all of these challenges? I hope that you remember at least some part of what we did, right? All the different projects, human genome projects, HapMap project, and all of those different things, right? Then we moved to structures. We actually went through why study protein structures and how many structures are there. Again, omics, structural genomics initiatives, basic properties of amino acids, folding, elements of a protein structures, and all these structural motifs, right? I hope it rings a bell. How important structure is to understand functions, right? Then we did a hands-on session on UniProt. We did, NCBI. We went through Genome-Wide Association catalog. I'm sure it all rings a bell for you, right? So, and then of course we did these yesterday, right? Structures, protein data bank, and I'm sure if I wake you in the middle of your sleep to ask you what are the universal resources for protein sequence information, what would you say? What is the universal hub for bioinformatics for sequences? What resources comes to your mind? PDB, yes, that is for structural information. PDB, yes, absolutely. PDB is for structural information. For if I ask you for protein sequence information, where would you go? What are the universal hubs for protein sequence information. UniProt, that's absolutely correct. And one other resource that we did, which is a universal hub, UniProt is correct, but there is one other resource, NCBI, absolutely correct. So fantastic. So you know NCBI, UniProt, if you know these two resources, of course, you know there's tons and tons of data that you can do and mine and use some of the resources that you've actually learned to do all of these things, right? So now I am going to spend a few minutes to ask you, are there any concepts or questions that you have that was confusing? We did, you know, very limited amount. Of course, there were, you know, there was lots of data, there's lots of things that you learned, but is there anything that was confusing, or is that something that you think that you're taking home, at least some aspect of what we talked about this whole week, right? Are there any specific questions that you think was important that it just went over your head, that, you know, you just didn't understand? I'm just going to spend a few minutes before I'm going to show you the activity that I have planned for you, right? So any questions at all that you have? I'm just going to wait for a minute and see if there are any concepts or something that actually, oh, I see, Ramachandran plot, that's correct. Thank you. So let me see whether I can actually, yes, I promised that and I forgot. Thanks for the reminder. Let me actually pull up something. Actually, it's one of the most important fundamental concepts of structural biology is actually Ramachandran plot, right? So I think I have, I had put it for yesterday, and let me see. I'm going to share my screen here. So this is pretty much what is the summary of all the scope that we just did, right? So we just did that. Yeah, so Ramachandran plot. So what do you know about Ramachandran plot? Have you all heard about Ramachandran plot? So what is the context in which, no, you have not heard about Ramachandran plot? Is anyone else has actually heard about Ramachandran plot? No, okay, so there are lots of no's, which is actually, okay, Suparna, you've heard about it, yes. So there are lots of no's of Ramachandran plot, and yeah, some of you have actually heard. How do you spell that? That's actually a, it's actually interesting. I'm going to show it to you in a second. It's one of those Indian names that's hard to spell. What is Ramachandran plot? It is actually fundamental to structural biology. And yes, I'm going to give you the spelling very, very soon. Hold on for 1 second, right? We're going to see. It's actually fundamental. Without the Ramachandran plot, no structural biologist can actually publish their structure. It's one of the most fundamental plot of, you all know, right? This is a peptide chain, and you already know that this is the amino terminus and this is the carboxy terminus. This is actually the peptide bond, right? NHCO, right? NHCOO, right? This is the peptide bond. And the fundamental principle is that the peptide bond that I've actually told you is planar. Peptide bond is planar, right? So there are two angles that define the peptide bond, right? So one is the phi angle. This is the peptide bond, right? So, and then there are two angles. I'm just going to see. So there are two angles of rotation about this peptide bond, one here and one here, right? There are two rotations around these bonds, which is a dihedral angle, which means it's the angle between around this bond, but an angle between these four atoms, right? CO, C double on O, NH. So this is an angle around this angle, and there's a rotation about this angle. These are the two angles that are called phi angle and the psi angle, right? So these are the most important angles in any protein structures. So anytime you talk about structures, you need to understand what are these two angles, phi and psi. That actually define that there is specific rotation that is possible, that goes with the geometrical constraints that a peptide bond is always a planar. So Ramachandran plot is phi is a dihedral angle around this bond, NC alpha, and psi is for same, this is for hetero, is a rotation around the C alpha C bond. This is fundamental to any structures. And the peptide bond is actually planar. There is nothing more to it, right? Now, what Ramachandran plot is, right? There are limitations around those phi and psi angle. And this is the side chain that has, so there are restrictions about. So as I've told you, right, every structure has to obey the law of physics. So you cannot have any rotation, any bond length that is allowed. It's not allowed. Everything follows some rules and laws, right? So what is Ramachandran plot? This is the person, and I really wish that he won the Nobel Prize, because every single structure actually has to go through this plot, right? And I'm honored to say, sitting here, and it may sound a little bragging here, I actually did my master's from this department of Ramachandran. Ramachandran, of course, he was beyond my time, but I can say that my uncle, maternal uncle, actually was his first student who actually came up with the technology of anomalous dispersion, and actually came up. He did not come up with the I mean, he actually directed the idea of phi and psi, but actually it was another student of Ramachandran Sasishekharan who came up with this plot. So it is actually a fundamental plot where it tells you that everything in the structure space follows rules. So which means there are only certain phi and psi angles that are allowed for a protein to actually have, right? So this area that you see in orange, right? So this is called the allowed region of the Ramachandran plot. And anywhere else is disallowed region of the Ramachandran plot, which means a protein cannot, an amino acid and a peptide bond cannot have any angle that it actually chooses to have. There are restrictions of rotations about the main chain angles. So they can only be, every single amino acid in a protein can only occupy any one of these spaces. There is only one exception to this rule. There is one amino acid that doesn't have to follow any of these rules. Which amino acid do you think that would be? That doesn't have to follow this rule of being anywhere only here in the shaded areas of allowed regions of Ramachandran plot. That particular amino acid can be anywhere else. Other than that, all the other 19 amino acids have to actually follow those rules. Which amino acid do you think doesn't have to follow rules? Not proline. Proline actually follows rules. Glycine, absolutely correct. Right? Glycine doesn't, because because it has it's tiny. It follows. It has only a hydrogen as a side chain. We discussed this, so it can actually be anywhere in this plot. Other than that, all other amino acid residues actually have to follow. Will actually have to obey these angles, phi and psi. And there is one more thing that they found was all helices actually will actually have. Here, these angles, anti-parallel beta sheet will actually have angles here. Parallel beta sheets, of course, I told you that parallel beta sheets is rare commodity in protein structures. You don't see it. And all alpha helices will have phi psi angles that fall here. That's it. This is the Ramachandran plot. It is divided into fully allowed regions and generously allowed the shaded regions, and completely disallowed region outside of these spaces is called disallowed region. So this is the Ramachandran plot. Glycine is highly flexible. It can actually occupy any one of these quadrants. Even though it is allowed to go anywhere, it still goes only somewhere here based on all the structures we have actually. So every single protein structure in protein data bank determined by X-ray crystallography, NMR, electron microscopy, we actually put this plot. Without this plot, and we have to make sure there are no amino acids in this disallowed regions. If there are amino acids in this disallowed regions, what does it mean? Now that I've explained to you what Ramachandran plot is, if I actually say that there is, if I see a lot of amino acids here in this white region, which it's not allowed to be, what does it mean if I see it? Yes, it means that the structure, those amino acids are actually, their geometry is wrong, is placed in the wrong position, and you have to go back and fix it, right? Now it doesn't say much about the stability. It just says more about the geometry, which means it is wrong. The stereochemistry and the angles of the amino acid is actually wrong. So which means we go back and fix it, right? We have to go back and fix it. Sometimes we may not have the data to fix it. So which means the structure, you cannot rely upon those structure, right? Those are wrong amino, they're placed wrong, right? But very rarely, sometimes those wrong positions may actually be exceptions to the rule, and you may be discovering something about that particular amino acid. So it does exist. So about 1 or 2% actually are usually allowed. This is actually the Ramachandran plot of my own structure that got me my PhD. So this is my Ramachandran plot, and you actually see that there are residues here that are placed wrong for me in my structure. But then you know what? I found that there is, whatever I do, it always comes. That means there is an interesting, I found some interesting geometrical implications for these amino acids, right? So it is not wrong. So usually about 2% of amino acid residues are actually allowed because data dictates the structure, right? Sometimes there could be problems with the data, there may be missing data and all of those things. So some amount of percentage of this is actually allowed. So that is, I'm glad you brought it up because this was my promise to you that I'll show you Ramachandran plot. So does it clarify? Do you understand what Ramachandran plot is? Yes, any questions? I know that we are running out of time. I have just a couple more minutes. If there are any questions, of course, you can, ask me, email or, contact, let Bipsha know, or I think there is a blog, right? You can post it there, and Bipsha can send those questions and I can answer those questions for you, right? So now what I am going to do is going to show you, it is the forum. Yes. Okay. So what I'm going to do is now very quickly, I am going to talk about the activity. I am not sure if Mohit, Ilya, do you have any comments about the activity? Do you think, I don't know if you got a chance to see the activity. I had emailed it, but if not, so this is the activity that I have for you guys. I'm sure it's, I don't know if it's, I think Bipsha, it's available to them, correct?
I have not sent it to them yet. I'll be sending.
Okay, sure. So thank you though. It's good you did not send it, so that I think after my explanation will make more sense. Thank you for not sending it actually.
Yeah.
So now what I want you all to do is please pick any disease that is interest of you, that is of interest to you, or you can pick any one of these, right? How would you go to pick a disease of interest to you? So we have actually done the hands-on, correct? Which database would you go through? And I'm sure you all know how to do that, right? You can go, we did that in the UniProt hands-on. I also showed you in PDB, there is lots of diseases, that you can pick from. And I actually honestly would pick from, I think Jeffrey had asked this question, how do you pick something that's very interesting for research purposes? I would actually pick a protein that's implicated in a disease. You can start with UniProt, do your advanced search, pick something that actually has a 3-dimensional structure, and so that you can do a full-fledged thing of practice of all that we have talked about this week, correct? So you can actually, so this is one. So I've actually come up with some questions, specific questions for you, so that you will learn how to use the resource well with the example that you actually, and then you can write these answers in a Word document, or Mohit, I don't know if you have a comment about this or any particular format. You can answer these, or you can actually prepare this in a form of a presentation. You can present it in PowerPoint. You can put it in a form of a PowerPoint. Classify, group some of the answers, and you can categorize them. And if you were to give a presentation about that particular disease that you are actually picking and all the data that you've collected, you can just put it together as a PowerPoint presentation of like maybe maximum of 3 to 5 slides. And then, of course, you can use all of these resources. These are the questions that you'll use: PDB, SCOP, and PyMol. And then, of course, this is something that I really...
Sona, I just wanted to insert one quick comment. Sure. Like about the format, it's just simply that you guys have to use a PowerPoint presentation.
Okay.
Put your things in there and try to, if possible, try to record it. Like if you can record it and send it, then we will be able to, learn that what you have done from that. So that would be helpful.
Sure, that's a fantastic idea. I think you can answer all these questions and then put them as a presentation. How would you present to me or Mohit or Ilya or Bipsha? How would you present this, or the judges? How would you present this to someone who...
Yeah, that's why I'm saying that they can record it, right? They can record it. So yeah, you can see, listen to it. Yeah.
Okay, sure. Okay, so there is a, can you show the previous slide, please? Okay. Interest in polycystic kidney disease, but I can't figure out what mutations cause cystic form. What should so I think, Kylie, you can actually go to UniProt and use this particular disease you're interested in as the search. And then from that, I'm sure that whole page of under natural variants in that UniProt that we actually looked at, you can just look at that, and also look at publications that are associated with it. So maybe there is something in the literature where they've actually come up with the specific polymorphisms. Will these yeah, of course, these instructions will be available to you all. As soon as Abhipsha is waiting for me to walk you through so that, you don't get lost before, you don't go ahead and get lost, right? So, okay, so now, yes, as Mohit said, I think it'll be a fantastic idea that he said, that, you put it as a part of a PowerPoint presentation, record yourself, and actually group all the questions that I've placed here and just make it a presentation, right? You can group saying, what is the function of this? What is the disease? Write about the disease, give an introduction, give, you can organize this in your own way so that you're giving a presentation, right? And then this is something really, really fun that you can actually, I think I showed you this as we did the hands-on, that you can actually build paper models of any one of the molecules that's out there, insulin, tRNA. It's really, really, really fun to build models. I am not going to mandate this, but if you build a model, you can bring it to class next week and we can actually, you can show it to us or take a picture and put it in your PowerPoint slide as one of your slides. You can take a picture of your model and put it there, right? So, so that is it from me. And since we don't have much time, I hope that you all felt you learned at least something from this whole week. I hope it was useful. It was, something that you can carry home with you. And for me, it was a pleasure to be with you this whole week. And I think I'll miss this opportunity, but I'll join next week on and off because we still have the whole week next week. I'll join on and off. And anyway, so I'm not going away. You're still going to see me at some point. And so that's it from me. Any other of this reference of the, yes, you will all receive this. Yes. Absolutely. You'll all receive all of this information from Bipsha. Yeah, thank you guys. Thank you. Thank you.
Thank you so much, Dr. Sona. It was
yes.
lovely having you here and we learned so much from you. Thank you for sharing.
Thank you. Thank you so much. Thank you guys. Thank you all for being patient and,
thank you, Sona. Yeah. And for next week, we will pick up from where we left off here, and hopefully you, so also about these presentations and the recordings, don't feel like you have to have everything, final and polished before you let us see what you have. We are here to comment on, any progress that you make between now and the end of next week, but we hope to see something before then.
And if I may just let everybody know, I will be emailing you all of these details, including the PPT, so you do not have to worry about anything. And you can reach out to me and ask if you are missing any resource or if you do not have anything, because I've sent you all the resources. But in case you have not received those emails, then just let me know, okay? marketing@pine.bio, or you can even post it on the forum that, I'm not understanding this. And as you see that we are answering all of your questions that you're having regarding the courses on the forum. So keep doing that. You can post in either of the places, either emailing me or on the forum. And thank you so much, everyone. Thank you for attending today. Thank you, Dr. Sona again. Thank you, Ilya and Mohit. And also Dr. Iyer is not here, but thanks to him as well for the wonderful session.
Yes, thank you, Bipsha.
Thank
and
you
I just
all.
wanted to add
go ahead.
just one thing, that you all have access to the lectures that Dr. Sona gave. So in case you are finding an issue while doing some analysis, you can go back to that video and see how it was done. So just in case, all those videos are available in the forum and within your emails. So look for those as well.
Okay, so good night, good morning, good evening, wherever you are. Have a good weekend, and we'll be in touch.
Have a good weekend. Thank you.
Yeah, bye-bye.